Now that you have some practice doing some DNA to DNA problems, kind of replicating what DNA replication would look like, if you have template DNA, you can figure out what the coding DNA, the opposite strand of DNA, would look like. And you've done some DNA to RNA problems, some transcription type problems, where if I gave you a segment of DNA, you would be able to tell me what the mRNA would look like that came from that DNA. So here I have a ribbon of mRNA that I've made up. You can read the nucleotides and see that it's RNA. You got AUG, UGG, UUU, AUG, UGG, UAA. And if I asked you, you could use that chart, that genetic code chart that we used last time. It's in your textbook and it's in your notes in order to translate this message out. So you understand that that's what the ribosome does, right? Ribosomes do the work of translation. They take that message and they figure out what amino acids go in which order in the polypeptide that they are making. But they don't have a chart. They don't have a reference chart. So how does the ribosome know that AUG means methionine, for instance? How is the ribosome so good at this particular job when it doesn't have the ability to read, it doesn't have the ability to think? Well, the secret here is that there's multiple kinds of RNA, and we need several of them in order to be able to do this job. It shouldn't be shocking that there's multiple kinds of RNA because we've already seen two of them. Two kinds of RNA. When we were doing RNA processing, we ran into a kind of RNA that was used to splice the exons together, right? Splice those coding bits of RNA together. That is called snRNA. That's snRNA. This message here that we're going to translate out, that's mRNA. But in order to translate this message, we need to go to the ribosome. And the ribosome is also made out of RNA. In fact, all of the functional parts of the ribosome are made out of RNA. The ribosome is essentially one big enzyme. It does the work of catalyzing dehydration reactions between amino acids. So it helps to synthesize those polypeptides. But the functional part of that ribosome is RNA. So the ribosome is actually a ribozyme. We call an RNA enzyme a ribozyme. So the ribosome is made of another kind of RNA, and that's called rRNA, ribosomal. Ribosome RNA. Right? We have messenger RNA, we have ribosome RNA, and then in order to translate this message, we're going to need something else, and it's called transfer RNA. But in my head and in my heart, I like to think of it as translation RNA, and it's called tRNA. tRNAs are our molecular interpreters. Okay, so they're actually called transfer RNAs. Transfer RNAs. And what they do is they carry amino acids around. Every tRNA has one particular amino acid that it's kind of shuttling around here. So here I have one of my tRNAs. Right here is a tRNA that's kind of floating around outside of the ribosome, carrying an amino acid, in this case phenylalanine, and it's waiting for its opportunity to shine so that it could come in and deliver this amino acid to the growing polypeptide chain. But it can't until it's actually its turn. Every tRNA has two important sections to it. It's got the amino acid attachment site, where it will be attached to whatever amino acid it's carrying around, and then it's got this section down here. And this is where the mRNA message gets read. See these little bumps and grooves here? This is a sequence of 3 RNA bases. A sequence of 3 RNA bases, we've heard of that before. That's a codon. Every 3 letters is a codon, right? The codons appear on the mRNA. But the tRNA has what we call anticodons, because these are meant to pair up with the mRNA codon. So mRNA has codons, tRNAs have anticodons. Now, if you take a look at this slide right here, you can see how the tRNAs and the ribosome work together in order to translate that mRNA message. The mRNA comes into the ribosome and it gets locked into place. Okay? The ribosome is basically a workbench for making polypeptides. And it's got these three little docking stations on it. You see those three little grooves on the ribosome? Those are where the tRNAs are allowed to come in. So in the cell, we have all these tRNAs just kind of floating around, minding their own business, each one with its own amino acid attached to it. And then they have this anticodon that can read mRNA. And periodically, they float into these little chambers on the ribosome. And if their anticodon matches up with the mRNA codon, then that's good. They can actually deliver their amino acid to the growing polypeptide chain at that point. So transfer RNA is the molecular interpreter. This is what's actually able to read the mRNA message, and this is what brings the correct amino acid into that position. Here's an analogy for thinking about translating using all these tRNAs. Imagine I said, you know what? Your biology students, you should learn how to speak Greek because we have all these Greek words that we use in biology. You're all going to learn how to speak Greek. And I'm going to give you a test on Greek. I'm going to put up a 1-paragraph poem in front of the class, and I want you all to translate it. So that will be your test on Greek. And you think that's insane. When are you going to do this? I say, I'm going to do it tomorrow. You're going to have to translate this tomorrow. Now, that would be very difficult if I expected each student to translate the entire message. But what if instead I said, every student will be responsible for only one word. You need to recognize a single word in that poem, and when you see that word, you can write the translation for that whole word. And then all of the students together can work together in order to translate the poem. If we did that, it would be very, very simple to be able to translate the message. So this is how tRNAs work. Every tRNA has a different anticodon, so it recognizes a different segment of mRNA, and it has a different amino acid attached to it. Now, some of them, because some codons code for the same thing, will have the same amino acids, but there will be an anticodon for every possible combination of nucleotide bases that we could have on an mRNA molecule. Now, on these slides, they use a kind of image of tRNAs that kind of look like my papercraft image right here. It's got the important bits on it. You got the amino acid attachment site up here, and you got what we call the anticodon down here. But if you actually want to know what a tRNA molecule looks like, that's it right there. After all, RNA is a single-stranded molecule. You can see that this is a single strand that's actually wrapped back upon itself a couple times, and it's hydrogen bonding with itself in a couple locations. So that's what the structure of a tRNA molecule is, but for most purposes, this is a simple enough model to get the job done. You have the anticodon and you have the amino acid attachment site. Those are the important parts of the tRNA molecule. Now, the ribosome is actually made of two different parts. They're called subunits. You have the large ribosomal subunit and the small ribosomal subunit. The small ribosomal subunit kind of holds the mRNA in place. It attaches it and it helps it to kind of move down the line as we are translating. The large ribosomal subunit contains these docking sites for the tRNAs to be able to enter. So you can see these little pockets. It has the A site and the P site on it. There's actually three. There's an E site to the left of the P site, but it's not on this diagram for some reason. Ribosomes are made out of protein, which is kind of a scaffolding to hold them together, and then the functional parts of them are made out of rRNA, ribosomal RNA. So we have multiple kinds of RNA involved in this translation process. Way back when we talked about DNA and RNA, I said that DNA is really good at carrying information, and RNA has many, many tasks inside the cell. I was a little bit vague about it at the time because it has such an interesting diversity of tasks that it's really hard to nail down. They're kind of like proteins in that way. They can do lots of different things. They can catalyze reactions, they can carry information, they can replicate themselves. RNA are very interesting molecules. What you can see here is that each tRNA comes in and it tries to match up its anticodon, its message reading component with the mRNA codons. And if it gets a match, the amino acid that it's carrying is going to be added to that chain. Once it gets added, the whole thing moves down 3 letters, and the previous tRNA that was holding the polypeptide chain gets to leave, and the new polypeptide that just entered carries that new chain that it's hanging on to here. So we'll see this kind of in process. Now there are 3 steps of translation, and the good news is that the titles of these 3 steps are the same words we use to talk about the 3 steps of transcription. Remember what those were? Initiation, elongation, termination. But this means that I can ask tricky questions. I could say something like, in the initiation step of transcription, and then give you a multiple choice question that includes answers for both transcription and translation. You would need to know what happens in transcription versus translation. So pay attention to these parts. Just knowing the titles is a good start, but you're going to need to know more detail. In the initiation step of translation, we need to bring some of our components together. What do we need to bring together? We got the large ribosomal subunit and the small ribosomal subunit. We have the mRNA that we're going to be translating. We have all those tRNAs. They all got to come together during the initiation phase to get this party started. Here you can see an mRNA ribbon, a mRNA string, that is fully processed. You can tell because it's got the 5' cap and it's got the poly A tail on the 3' end there. We know that it's already been processed. All of the introns have been cut out. All of the exons have been spliced together. We're going to bring it in for the first step of translation, which is initiation. During initiation, the small ribosomal subunit is going to attach itself to the mRNA message here, to the mRNA ribbon. Then the large ribosomal subunit is going to clamp down on it. Now, the first site on the large ribosomal subunit is called the A site. So a tRNA is going to come in, and it's going to try to match its anticodons to the mRNA codons. You can see that image kind of zoomed in the center of the slide here. If we have a match, so for example, this mRNA has AUG for the first 3 letters, that's the first codon, the anticodon would therefore have to read UAC in order to match up with that. This is RNA to RNA, so there will be no T's here. We have 2 different RNA molecules, mRNA and tRNA. If we have a match, then that is your first amino acid in the polypeptide chain. Once we have a match, we can begin the next phase. So that's initiation. We brought together all 3 components here. We have the ribosome, large and small subunits, we have the mRNA, and we have the tRNAs with their amino acids coming in. Now, we begin the process of elongation. When we were talking about transcription, elongation was referring to elongating the mRNA ribbon, the mRNA string, the mRNA strand. But here, elongation refers to elongating the polypeptide, so adding more amino acids to this polypeptide chain here. Once we have initiation complete, the entire message moves down 3 letters. The ribosome just moves the mRNA down 3 letters, and it looks at the next codon. So we have a new codon available in the A site of the ribosome. Meanwhile, the first tRNA that came in, it's moved over to the P site. Next, another tRNA is going to enter the A site. It's going to match its anticodons to the mRNA codons, and if it has a good match, then its amino acid will be bound to the first amino acid, so we'll have a dipeptide at that point. Then the whole thing moves down another 3 letters, which we call translocation. And this process happens over and over and over again. So a new tRNA comes into the A site, it recognizes the codon there, it brings its amino acid in, and we catalyze a bond between the 2 amino acids. We catalyze a peptide bond. Every time we translocate the whole message down 3 letters, the A site opens up again for a new tRNA to come in, and then they can get a new peptide bond at the P site. The whole thing moves down 3 letters, new tRNAs come in. So tRNAs enter through the A site and they exit through the P site. Well, actually they exit through something called the E site. Now this diagram has it's got some nice pictures to it, but I think it's a little bit more helpful if I kind of perform it out for you. So enjoy this. The process of translation requires that several different components are going to come together. The large ribosomal subunit and the small ribosomal subunit. The large ribosomal subunit has 3 sites on it. These are the A, P, and E sites. New tRNAs enter through the A site, move to the P site, and then exit through the E site. The small ribosomal subunit is there in order to hold the mRNA molecule in place, and it locks in just underneath. When translation begins, the mRNA molecule will bind to the large and small ribosomal subunits, and a number of tRNAs that are just floating around inside of the cell are going to float into the A site and see if their anticodons match with the mRNA codons. Eventually, one of them will have the correct anticodon and will get a fit. Then the entire thing will translocate, it will move down 3 bases, and another tRNA will enter the A site. At this point, a peptide bond is formed between the first 2 amino acids. Once the tRNA's job is done, it exits through the E site, where an enzyme out in the cytosol will attach another amino acid to it, and an amino acid appropriate to its anticodon. New tRNAs will enter through the A site, the whole message will translocate down 3 letters, a new peptide bond will form, and then another anticodon will try to float into the A site again. This process continues over and over and over again, with new tRNAs entering through the A site and exiting through the E site, and new peptide bonds being formed at the P site until eventually a stop codon is reached. A stop codon is recognized by a tRNA which itself does not have an amino acid. The effect of this is that when that stop codon translocates down to the P site, no new peptide bond can be formed. And if no bond is formed, then when the whole thing moves down 3 letters, again, the entire polypeptide will be released from the ribosome. At this point, the mRNA and the large and small ribosomal subunits will uncouple from each other, and we have reached the final step of translation, which is called termination. Termination occurs when a stop codon is reached.